Welcome friends. In this video, I would like to discuss about tryptophan operon. This operon has two words. One is the tryptophan and the other is the operon. Tryptophan, it is an amino acid that is synthesized that is synthesized by the prokaryotic cells. It is an essential amino acids in eukaryotic cells, and those they cannot synthesize tryptophan, which the eukaryotic cells cannot synthesize tryptophan, but the prokaryotic cells they can synthesize tryptophan, and that is operated by the tryptophan operon. So it is the synthesis of the tryptophan which is managed, which is coordinated, which is controlled and operated by an operon, and that is known as tryptophan operon. So the prokaryotic cells can synthesize tryptophan, but the eukaryotic cells, there it is an essential amino acid and it cannot be synthesized by the eukaryotic cells. But the prokaryotic cells can synthesize tryptophan that is coordinated and operated by tryptophan operon. So tryptophan is an amino acid having the chemical formula C11H12 N2 and O2. This is the chemical structure of the tryptophan. It is having L amino group, L carboxyl group and a side chain indole and a polar molecule with non-polar aromatic beta carbon constituent. So this is the tryptophan, and the tryptophan is very important because it forms one of the integral amino acids in a number of proteins. The proteins which are synthesized having the tryptophan as amino acid, and it is a very important amino acid which is synthesized by the tryptophan operon. So the tryptophan operon is a negative. Tryptophan operon is a repressive negative repressive anabolic operon. It is a negative repressive anabolic operon. It is negative because here the operon is switched off by repressor and not by an activator. In positive operon, it is the activator which switches off and on the operon, but here it is negative because it is switched off by a repressor. And the repressor is along with a co-repressor, hence it is the repressive type of operon and is anabolic because this operon is for the synthesis of the tryptophan from chorismate. So the biosynthetic pathway of the tryptophan has these stages. One is the conversion of choristomate into anthranilate, and from anthranilate to 5-phosphosyl anthranilate, then carboxyphenylaminodeoxyribulose 5-phosphate, then indole 3-glycerol 3-phosphate, then indole, and this tryptophan. And all these stages are catalyzed by the enzymes such as the anthranilate synthase, anthranilate PR transferase, PRA isomerase, IGP synthase, 3-phosphoglyceraldehyde and tryptophan synthase. And these enzymes are encoded by the genes A, B, C, D, E, F, G. So the genes encoding these enzymes are A, B, C, D, E, F, G, but they are not in proper alphabetical sequence, and these genes are, the first conversion is encoded by gene E, second step by gene GD, third by gene F, and the fourth is gene C, fifth is gene A, and the sixth is gene B. So there are 6 genes A, B, C, D, E, F, G. They are encoding these enzymes. So the tryptophan operon means it is the operon, it is the genetic unit which is coordinating, which is operating the synthesis of tryptophan from the corysmate, and the enzymes which are synthesized are managed and encoded by the gene E, G, D, F, C, A and B. So the tryptophan operon is a negative repression anabolic operon. Now when there is a low level of tryptophan in the medium, when the medium is having the low level of, these are the structural genes having E, G, D, F, C, A and B. This is the operon. In the genetic unit, the structural genes are E, G, D, F, C, A and B. Then it is having an operator and a promoter, and like another operon, there is a regulator gene, and this regulator gene is synthesizing the repression. Now here the repression is aporepressure, and this aporepressure is, it remains inactive. It cannot bind itself to the operator. The repression is there in the medium, but it is inactive. The repression is inactive and it cannot bind itself to the operator. Hence, the RNA polymerase is free to carry on. It is free to carry on the process of gene expression. Gene is expressed normally when the level of tryptophan is low. The RNA polymerase is then free to carry on the process of gene expression. So when the tryptophan is low, when the tryptophan is low in the medium, the gene expression is on. Means there is a synthesis of all these, it is on, and all these enzymes are taking place. All these enzymes are synthesized, forming the messenger RNAs. Forming a messenger RNA with different types of protein. This is messenger RNA. It is polycystonic, hence it is capable of producing the different types of enzymes or the proteins, and the gene expression is on. When the tryptophan is low in the medium, the gene expression is on because the aporepressure is inactive. It is inactive and it cannot bind itself to the operator to stop gene expression. But when the tryptophan is high, in the medium, the gene expression is shut down. No gene expression taking place, and it is shut down, and the tryptophan itself, tryptophan acts as a co-repressor. The co-repressor is there. The tryptophan is acting as a co-repressor, and it changes the conformation of the repressor. The co-repressor, when it is attaching it to this, the repressor becomes active. The repressor co-repressor complex is then attached itself to the operator, and it is impossible for the RNA polymerase to carry on the process of gene expression. Means the gene expression is off, the gene expression is shut down, and there is no synthesis of, it is off. No further synthesis of tryptophan occurs. Here the tryptophan itself acting as a co-repressor. Tryptophan is the co-repressor, and this co-repressor aporepressor complex then shuts off the synthesis, the gene expression, and no more tryptophan is produced because the level of the tryptophan is high. So when the level of the tryptophan is low, the gene expression is on, and when it is high, then it is off. That is, it is closed, it is prevented, it is inhibited by the tryptophan, which is acting as co-repressor. This is a co-repressor along with the aporepressor. Co-repressor along with the aporepressor is now completely blocks gene expression, completely inhibits gene expression, and no more synthesis of the tryptophan takes place. So tryptophan is synthesized when there is a low level of tryptophan. So when there is a high level of tryptophan, there is no need of synthesis of more tryptophan. So the tryptophan here acting as a co-repressor to inhibits gene expression. There is one fine tuning is there with a set of genes known as leader, and this leader acts at the level of post transcription. Means when the transcription has already started, at that time the leader comes into action and it inhibits the gene expression, and that is described in the next video. That is known as the tryptophan attenuation. So the tryptophan operon is on when the level of the tryptophan is low. It is off when the level of the tryptophan is high. And this is an anabolic. This is an anabolic operon because it is the synthesis of the tryptophan from the chorestomate that is catalyzed by these enzymes, by these 6 enzymes, and these enzymes are encoded by these genes which are forming the structural genes in the tryptophan operon. So tryptophan operon is a negative repressive and anabolic operon. So the tryptophan attenuation I will describe in the next video. Thank you.